Okay, let's dive in. We're talking about critical limb ischemia today, CLI. That's the really serious end stage of peripheral artery disease.
Right, where the blood flow to the legs and feet gets dangerously low. We're talking tissue loss or constant pain even at rest. Serious stuff.
And a key treatment when it gets that bad is bypass surgery below the groin using one of the patient's own veins, right?
Exactly. An infra-inguinal bypass with an autogenous vein graft, it's often the best shot at restoring flow.
It sounds vital, but there's a catch, isn't there? These vein grafts,
Yeah,
they don't always go the distance.
that is the crucial challenge here. Yeah. Despite everything, a pretty significant number of these bypasses fail over time, often within just a few years.
Fail how? What actually happens?
Usually it's a narrowing inside the graft itself. We call it stenosis, and that, unfortunately, can mean more procedures, more problems for patients who are already dealing with a lot.
Okay, let's put some numbers on this. This isn't a rare operation, is it?
No, not at all. If you look at the Vascular Quality Initiative data, it suggests it's the first approach in maybe 37% of CLI cases.
Wow.
And a German registry had it around 24%. So yeah, a lot of people rely on this.
But then you see these failure rates. What do the sources say? 30 to 50% losing what they call primary patency within 5 years.
That's right, 30 to 50%. It means the graft isn't working on its own anymore. That's a major, major challenge.
So even with this critical surgery, the long-term benefit isn't guaranteed.
Exactly. And while we do try to keep an eye on them, regular ultrasound checks.
Surveillance.
Right, surveillance. And we can intervene if we spot problems early, try to keep the graft open longer. That's called assisted patency.
But that's not ideal either, is it?
Well, no. It adds cost, complexity, and more procedures for the patient. It doesn't fundamentally stop the underlying issue that causes the graft to narrow in the first place.
So the hunt has been on for a long time to find something better, something specific to make these vein grafts last longer.
Precisely. Beyond just the standard things we do for PAD generally, blood thinners, statins, lifestyle advice, we need something targeted at the graft itself.
And that brings us squarely to the PREVENT-3 trial. This was a big attempt to tackle that specific problem, wasn't it?
It really was. A large multi-center study looking at a pretty novel idea at the time.
Which was?
Could we use a targeted molecular therapy, a drug basically applied directly to the vein graft during the surgery?
Ah, okay. Treat the vein before it even goes in.
Exactly. The thinking was the vein is accessible right there on the table before it gets implanted and faces the stress of arterial blood flow. Maybe we could kind of pre-treat it, make it more resilient.
Okay, that makes sense. But why does the vein need protecting? What happens inside it after the surgery that causes this failure?
Well, it's a really complex biological response. Think about it. You're taking a vein which is used to low pressure, low flow, and suddenly you plummet into the high pressure, high flow arterial system. The vein reacts to that new environment.
How does it react?
It triggers a whole cascade. Inflammation, some clotting activation, and critically, cells within the vein wall start to migrate and multiply, especially smooth muscle cells.
They multiply.
Yeah, they proliferate, and the tissue around them changes too. It's sort of like an injury response, but it can get out of hand.
And just handling the vein during surgery causes some issues too, right?
Absolutely. The surgical process itself, harvesting the vein, manipulating it, it inevitably causes some initial damage. You lose some of the delicate inner lining, the endothelium, and injure other cells.
So it starts off slightly damaged and then gets this big shock from the arterial flow.
Pretty much, and almost immediately you see this surge in cell growth within the vein wall as it tries to cope. It's similar to what happens after, say, an angioplasty in an artery, but in vein grafts, this proliferative response seems to last longer.
But hang on, doesn't the vein need to thicken up a bit to handle the higher pressure?
That's a really sharp point. Yes, a certain amount of wall thickening is necessary.
Yeah.
It needs that biomechanical stability for its new job. The problem is when that thickening process goes too far, becomes excessive. That's what leads to the stenosis, the narrowing that chokes off blood flow and causes the graft to fail.
So the goal is to moderate that thickening, let it adapt, but not overdo it.
Precisely. Find that sweet spot. And that's where the PREVENT-3 intervention comes in.
Right. Etifologide. What exactly was this drug?
Etifologide. It's technically a double-stranded oligodeoxynucleotide, basically a short piece of synthetic DNA.
Okay. And what did it do?
It was designed to act as a decoy. It mimics the binding site for a key protein called E2F.
E2F.
Yeah, E2F is a transcription factor. Think of it like a master switch that turns on genes needed for cells to enter the cell cycle and divide.
So Edifologide basically jams that switch. It binds E2F so the E2F can't turn on those growth genes in the smooth muscle cells.
Exactly. It inhibits the E2F pathway. Lab studies showed it dramatically stopped vascular smooth muscle cells from multiplying, and importantly, with very little toxicity.
And did it work in living systems before they tried it in humans?
Yes, it showed real promise in animal models. For instance, in rabbit carotid artery vein grafts, it significantly reduced that problematic inner layer, thickening the neointimal hyperplasia.
That does sound promising. How did they actually get it onto the vein during surgery? Was it complicated?
No, actually, the delivery was quite elegant and simple. After the surgeon took out the vein, but before sewing it in, they'd place the vein segment into a small, sealed chamber. Then they'd fill the chamber with either the Edifologide solution or a placebo solution.
And just let it soak?
Pretty much, under gentle, non-distending pressure for about 15 minutes or so. A straightforward one-time treatment during the operation.
And there were earlier human studies suggesting this might work.
Yes, there were some smaller early-phase pilot studies in humans, both for leg bypasses and for coronary artery bypass graft.
What did those show?
Primarily, they showed it was safe and feasible to do this intraoperative treatment, but they also gave some encouraging hints, some signals that it might actually be reducing graft problems.
Okay, so the stage was set. Promising mechanism, animal data, early human signals.
Right, and this led a biotech startup, Tech, to team up with a big pharma company, Bristol Myers Squibb.
To go for the big leagues.
Exactly. To run the large phase 3 trials needed for FDA approval, Prevent 3 for the leg bypasses, and a parallel trial, Prevent 4 for coronary bypasses. The goal was ambitious, potentially establish a whole new standard of care.
A lot riding on it. So let's dig into Prevent 3's design. Huge study, right?
Massive. They enrolled 1,404 patients who had critical limb ischemia.
Wow.
Across 83 different hospitals in North America over about 2 years. It was a major collaboration between academic centers and community surgeons.
Which is good. Means the results should apply more broadly.
Definitely. And the criteria for who got into the study were quite broad, basically mirroring the real-world clinical definition of CLI patients with tissue loss, like ulcers or gangrene, or those with severe pain at rest, plus objective proof of poor circulation.
What about the grafts themselves? Was it just a standard leg vein?
They allowed any autogenous vein, so the patient's own vein. The great saphenous vein was most common, used in about 80% of cases, as you'd expect, but they also included cases where surgeons had to splice veins together, or use veins from the arm. And interestingly, about 16% of the operations were redo bypasses.
Meaning the patient had already had a bypass fail before.
Correct, which again tells you something about the complexity and the high-risk nature of the patients they were studying.
Can you paint a picture of the typical patient in this trial?
Yeah, they were dealing with very advanced disease. 75% had actual tissue loss when they enrolled. Nearly 2/3, 64%, had diabetes, and 12% were on dialysis because of kidney failure.
A very sick group.
Very. And the bypasses themselves were often challenging, needing to go far down the leg. Over half, 53%, went to the tibial arteries just above the ankle, and another 12% went even further, down to the pedal arteries in the foot itself.
Really pushing the limits of revascularization. Okay, so they do the surgery, apply the drug or placebo. How do they track what happened next?
They had a very rigorous follow-up protocol. Everyone got duplex ultrasounds of their graft at specific times, 1 month, 3 months, 6 months, and 12 months post-op.
To look for that narrowing.
Exactly, to look for developing stenosis or other problems. And the study clearly defined when an intervention was needed, like if symptoms came back, or if imaging showed significant narrowing, defined as over 70% on an angiogram, or classified as severe based on the ultrasound flow velocities.
Got it. And the main thing they measured, the primary endpoint, what was that specifically?
The primary efficacy endpoint was the time until the first occurrence of what they termed non-technical index graft failure within 12 months.
Okay, break that down. Non-technical index graft failure.
Right. Index graft just means the bypass graft put in during the study surgery. Failure meant either the patient needed another procedure, like an angioplasty or surgery on that graft, or they ended up needing a major amputation of that limb because the graft wasn't working.
And non-technical. Why exclude technical failures?
That was crucial. They wanted to isolate the biological effect of the drug on the vein graft itself, not penalize it for, say, a stitch that came loose or some other surgical mishap unrelated to the vein's biology.
Makes sense.
So they had a blinded committee of experts review every single potential failure event. These experts didn't know if the patient got Edifologide or placebo, and they decided if the failure was truly due to the graft itself developing a problem or if it was down to a technical issue with the surgery.
Okay, a very careful definition.
Yeah.
So after all that work, enrolling over 1,400 high-risk patients, treating them, following them closely for a year,
Wow.
what was the big result? Did Etifologide work?
Well, the primary finding was
Yeah.
no, there was no statistically significant difference in that main outcome.
Really? No difference at all?
Essentially none. The rate of non-technical graft failure was 25.2% in the Etifologide group and 25.5% in the placebo group. Virtually identical.
Wow. So the headline is, this promising targeted molecular therapy didn't actually prevent graft failure as hoped.
That was the main conclusion, yes. A definite disappointment given the preclinical work and pilot studies.
What about other measures? Did they look at anything else? Secondary endpoints?
They did. They looked at things like overall clinical failure, including technical failures, whether grafts develop significant stenosis, and a combined measure of survival without needing amputation or reintervention.
Indeed, difference there.
Again, for most of these secondary endpoints, no significant differences emerged between the Etifologide and placebo groups.
But the study did provide some useful numbers on overall outcomes, didn't it? Like general patency rates.
Absolutely. For the entire cohort, the one-year results were quite informative. Primary patency, the graft working without any intervention, was 61%.
Right.
Primary assisted patency grafts kept open with interventions was 77%. And secondary patency, meaning grafts that failed but were successfully salvaged, was 81%.
Those numbers themselves are valuable benchmarks, I suppose.
Definitely. But there was one interesting finding in the secondary analyses related to patency.
Oh, what was that?
They found a statistically significant improvement in secondary patency in the group that got Etifologide. It was 83% versus 78% in the placebo group at 1 year.
So grafts treated with Edifologide were slightly more likely to be successfully reopened if they failed.
That seems to be the case. And intriguingly, when they looked closer, this difference seemed to mostly happen very early on, like within the first 30 days after surgery.
So maybe it helps stabilize things right at the beginning, making early failures less catastrophic or easier to fix.
That's one possible interpretation, yes. It's a subtle finding, not the primary goal, but interesting nonetheless.
And the overall outcomes for everyone in the trial: survival, amputation.
Also important benchmarks. At 1 year, overall mortality was low, 2.7%. Early graft failure within 30 days happened in about 5.2%. Survival was 84%, and 88% of patients avoided a major amputation on the treated leg.
So despite the graft durability issues, the overall 1-year limb salvage and survival were pretty good for such a sick population.
Yes, these figures really set a standard for what could be achieved with infra-inguinal bypass in CLI patients at that time.
Now, you mentioned a parallel trial for heart bypasses, PREVENT-4. Did Etifologide work there?
Unfortunately, no. PREVENT-4 involved about 3,000 patients getting coronary artery bypass grafts, and the results were similarly negative. Etifologide didn't reduce vein graft failure in the heart setting either.
So a double disappointment for this drug.
It was, yes. The initial hope for Etifologide as a broad solution for vein graft failure didn't pan out in these large trials.
But it sounds like you're saying these trials weren't a total loss, they still had value.
Oh, absolutely. Even with the negative result for the drug itself, PREVENT-3 and 4 were landmark studies. Just the scale of them investigating an intraoperative molecular therapy in cardiovascular surgery was significant.
How so?
They generated an enormous amount of high-quality data on how these bypass procedures are done in the real world, both academic and community settings, and what the outcomes actually are for these complex patients. They provided invaluable benchmark data.
Benchmarks for future research.
Exactly. Any new therapy or approach trying to improve bypass outcomes will inevitably be compared against the results seen in the PREVENT trials. They set a baseline.
And I gather the researchers didn't just stop with the main drug result. They dug deeper into the PREVENT-3 data.
They did. The richness of the dataset allowed for numerous secondary analyses, pre-planned ones, that gave us some really important insights.
Like what? What else did we learn from PREVENT-3?
Several things. For example, the analyses strongly reinforced just how crucial using a good quality, great saphenous vein is for long-term graft success. That might sound obvious, but this large data set really quantified it.
Okay, vein quality matters. What else?
They looked at how graft problems, needing reinterventions, impacted patients' quality of life. They compared the durability of fixing failing grafts with surgery versus less invasive endovascular techniques. They even found some associations between patient factors like race and gender and the risk of graft failure, prompting further investigation.
So lots of useful clinical correlations came out of it.
Yes. And they also developed something called the PI risk score.
PI risk score.
Yeah, using the PREVENT-3 data, they created a model to predict a patient's risk of amputation or death within a year after their revascularization procedure for CLI.
Is that actually used?
It's been validated in other patient groups, so yes, it has shown potential as a tool to help clinicians assess risk and perhaps guide treatment choices. The data from the placebo group has even been used to help set performance goals for newer endovascular treatments for CLI.
Fascinating. So even though the drug didn't work, the trial itself became a really valuable resource.
Immensely valuable.
But what about the big picture limitation? Did the study tell us why the grafts failed or why Edifologide didn't stop it?
That's the major remaining question, really. The study, by its nature, couldn't pinpoint the exact mechanisms. Vein graft failure is just so complex. Patient factors, the anatomy, the surgery itself, the underlying biology. It's multifactorial. Etifologide only targeted one pathway, the E2F cell cycle pathway. Maybe that just wasn't enough or wasn't the most critical pathway.
So where does that leave us? What's the next step in trying to solve this vein graft problem?
Well, future research probably needs different tools, maybe much higher resolution imaging to see subtle changes in the graft wall earlier.
Yeah.
Definitely deeper dives into the biology using modern omics approaches, genomics, proteomics, metabolomics, to get a much more detailed molecular picture of what's going wrong.
Looking beyond just cell proliferation.
Exactly. Understanding the role of inflammation, the coagulation system, different types of immune cells, like specific leukocyte phenotypes. We need a more holistic view.
So despite PREVENT-3, a good vein bypass is still a cornerstone treatment for advanced PED.
No, absolutely. When done well for the right patient, it remains arguably the most durable way to restore blood flow to the lower leg and foot in severe cases.
Blood.
But graft disease, that tendency for them to narrow and fail over time, is still a huge, largely unsolved problem. It significantly impacts patients' long-term outcomes and quality of life.
So the core idea behind PREVENT-3, trying to actively improve the vein graft with a targeted strategy, that idea is still relevant.
Very much so. The specific drug failed, but the concept of engineering a better, more resilient vein graft remains incredibly compelling. And here we are, 2 decades later, still searching for effective ways to do that. The quest continues.
Right, so wrapping this up, PREVENT-3 was a huge, ambitious trial. Etifologide didn't turn out to be the answer for preventing vein graft failure,
No, it didn't.
but the trial itself was far from a failure. It gave us vital benchmark data for CLI bypass outcomes, spurred numerous other important findings through secondary analyses, and really highlighted the sheer complexity of vein graft biology.
Exactly. It underscored that this isn't a simple problem with a single magic bullet solution. Improving long-term graft success is likely going to require understanding multiple pathways and maybe even combination therapies down the line.
It really drives home the challenges, but also the persistence in medical research, which leads to a final thought for you, our listener. Given these ongoing challenges with vein grafts, what other biological targets or therapeutic strategies might hold the key? Thinking beyond just cell proliferation, what other aspects of that complex vein response could we potentially modulate to finally make these vital conduits last longer? It certainly makes you appreciate the intricate puzzle researchers are still trying to solve.